our same chapter, pollination and fertilization. In our previous classes, we have learned about pollination, the types of pollination, the advantage of self-pollination, cross-pollination. Now today in our class, we will learn about the topic fertilization. So what is fertilization? We have learned that in pollination, just try to recall it. In pollination, there will be transfer of pollen grains from the anther of a flower to the stigma of the same flower or to another flower present on the same plant or on a different plant. Clear? So now after pollination, after the transfer of pollen grains, now there must be the process of fertilization where the pollen grain, upon reaching the stigma, they have to germinate and they have to fuse with the female gamete to ensure fertilization. Clear? So today we will discuss about fertilization. So let's begin. What is fertilization? Fertilization is the fusion of the male gamete and the female gamete. Clear? Fertilization is the process where the male gamete and the female gamete will be fused and results in the formation of a zygote. That we'll learn in detail when we go through the lesson. Clear? So, in previous class we learned that when the pollen grain germinates, clear? When the pollen grain germinates, that inside the pollen tube we will find two sperm nuclei or two male gametes that will help in fertilization. So now we will learn about the structure of pollen grain first. Okay, so firstly we will go through the structure of the pollen grain. So look at the board over here. So the structure of the pollen grain. Now when we look into the pollen grain, we know that it is composed of a double membrane structure. It's a double layer structure. There are two membranes. Okay, so there are two layers on the structure of the pollen grain. The outer layer of the pollen grain is called the exine. So you can see over here. So this is the outer layer of the pollen grain. So the outer layer of the pollen grain is called the exine, and the inner layer of the pollen grain is called the intine. Okay. So this is the outer layer. So these outer layer are called the exine, and the inner layer of the pollen grain, they are known as intine. So exine usually covers the entire pollen grain, but at a particular point, a small gap where exines are absent. So this particular region where exines are absent, they are called germ pore. Okay, so this particular regions where the exines are absent, they are called germ pore, and through the germ pore, the pollen tube will emerge. Clear? So pollen grain, it's a double layer structure. The outer layer of the pollen grain is called the exine, and the inner layer is called the intine. And in the outer layer where the exines are absent, it is called germ pore, and through the germ pore the pollen tube emerges or they will come. Clear? Now next thing, we'll go here. So inside the pollen grain, what we find? We'll find two types of nucleus: tube nucleus and generative nucleus. Clear? So inside the pollen grain we will find the tube nucleus and we will find generative nucleus. Clear this one? So look over here. Nucleus will be in two cell stages. The first cell stage is generative nucleus and the second one is tube nucleus. Alright? Next thing now. So after the pollen grain will mature, alright, after maturation, the pollen grain has to be transferred from the anther to the stigma. So maybe at this stage, when the pollen grain contains tube nucleus and generative nucleus, it will be transferred to the stigma, which is called pollination. And when the pollen grain reaches the stigma, it will germinate. Clear? So on germination through the germ pore, the pollen tube arises. Can you see over here? This is the pollen tube. And pollen tube will be formed only when the pollen grain germinates on reaching the stigma. And for that also, the pollen grain must belong to the same species. Clear? So when the pollen grain reaches the stigma and when it germinates, the pollen tube will be formed. And this pollen tube will come out through the germ pore. Clear? Now inside the pollen tube, listen carefully. Inside the pollen tube, we will find generative nucleus and we will find tube nucleus. Yeah. So inside the pollen tube, what will be present? We will find the generative nucleus and the tube nucleus. Remember, they are in two cell stages. Clear? And furthermore, this generative nucleus, alright, furthermore, this generative nucleus will again divide to form the two sperm nuclei. Can you see over here? So the generative nucleus will divide to form two sperm nuclei. We also call it male gametes. So at this point we can say that the pollen grains are in three cell stages. Why do we call it three cell stages? Because there are two sperm nucleus or nuclei and one tube nucleus. So 2 plus 1 will be 3. Clear? So this one is in three cell stage where we will find two sperm nuclei or two male gametes and one tube nucleus. Clear this one? So one more time, what you have learned? The pollen grain is a double layer structure. The outer layer is called the exine, the inner layer is called the intine. And the outer layer where the exines are absent, they are called germ pore, and through the germ pore the pollen tube emerges. So on germination, when the pollen grain reaches the stigma and when it germinates, the pollen tube will come out through the germ pore. Clear? And inside the pollen grain we will find two nucleus, generative nucleus and the tube nucleus. This is called two cell stage. And even in the pollen tube we will find the generative nucleus and the tube nucleus. And ultimately this generative nucleus will divide to form two sperm nuclei. So this is called the three cell stage with two sperm nuclei and with one tube nucleus. So these two sperm nuclei will help in fertilization, will help in diffusion of gametes. Clear? So these two sperm nuclei are very, very important to ensure fertilization in case of flowers. I have cleared for you the pollen grains. Now let's go to the next slide. Over here we will begin with ovule. Clear? We have finished the structure of the pollen grains. Now we will see the structure of ovule. Clear? So you can see over here this is the structure of ovule, an ovule which is present inside the ovary. Clear? So let's see what is ovule. Now ovule generally lead to the formation of the female gamete. The male gamete will call that two sperm nuclei produced by the pollen grain. Now ovule will lead to the formation of female gamete, which is called the egg. That will come afterwards. So when we look into the structure of the ovule, the female gamete, we see that the ovules are protected by an integuments. Clear? Though they have got name, we have got outer integument, we have got inner integument. You can see over here is the outer integument, the inner integuments. Clear? But just remember carefully in your syllabus, just go through that. The ovule is covered, is protected by an integument. So these are the integuments which will protect the ovule in its young stage. Clear? Now, so the entire ovule are surrounded or are covered by an integument. But you can see over here the small gap left over here. Can you see it? A small gap will be left over here where the integuments are absent. Clear? Generally the integuments cover or surrounds the ovule. It will ensure protection to the ovule. But at a particular point the integuments are absent. Clear? So this particular point where the integuments are absent, it is called micropyle. What do we call it? We call it micropyle. So you can look over here, micropyle, polytube inters. Clear? So what is micropyle actually? It is the particular particular point of the ovules where the integuments are absent. Clear? And through this micropyle, the pollen tubes enters inside the embryo sac. Can you see over here? This is the embryo sac. Clear? The embryo sac contains the egg which will help in fertilization. So through the micropyle, the pollen tube enters inside the embryo sac. So the structure of an ovule. So ovule is surrounded by a structure called integuments which will protect the ovules. At a particular point, the integuments are absent, and that region is called the micropyle. Through the micropyle, the pollen tubes enters inside the embryo sac. So this is the embryo sac, which we'll now talk in detail. So let's see the embryo sac. So you can see over here. So there is the structure of the embryo sac. If you get confused, please look over here. So now we are going to discuss about this part. Alright, this is the embryo sac. Now we are going to discuss about this part. So look over here. So when we look into the embryo sac, what we generally see is that embryo sac contains how many cells? It contains 7 cells. Clear? The embryo sac contains how many cells? It contains 7 cells. You can see over here the distribution of cells. 3 plus 3 plus 1. So it will be how many cells? 7 cells. So let's see the type of cells present inside the embryo sac. So you can see over here 1, 2, 3 cells. Opposite sides 1, 2, 3 cells. So 3 plus 3 will be 6. And at the center, look over here carefully, right? At the center, we will find 2 cells, right? How many cells are there? There are 2 cells. And when we count it, it will be 1, 2, 3, 4, 5, 6, 7, and 8. We are finding how many cells inside the embryo sac? We are finding 8 cells inside the embryo sac, right? But we are saying that the embryo sac contains how many cells? It contains 7 cells. Now though there are 8 cells inside the embryo sac, but why do we call it as 7 cells? Clear? Because, can you see over here? The central cell. Because the central cell, though it contains 2 nuclei, clear? It contains how many nuclei? It contains 2 nuclei, but it will be considered as a 1 cell. Clear? We have to consider it as a 1 cell. That's why we call it how many cells? 7 cells. 1, 2, 3, 4, 5, 6, and 1 will be 7. So this central cell is called the polar nuclei. It is formed by the fusion of 2 nuclei. So therefore, we will count it as a single cell. That's why inside the embryo sac we are finding how many cells? 7 cells. Now, next thing, remember carefully. The gametes, alright, the gametes, the male gametes and the female gametes, they are haploid, H-A-P-L-O-I-D. Haploid means containing half the number of chromosomes. Then, after fertilization, there will be formation of zygote. Zygote will be diploid to N. Or after fertilization, I am saying that when there will be fusion of gametes, zygote will be produced, which will be diploid, is called 2N. Now, for example, consider that we human beings, we have got 46 number of chromosomes. Alright, human beings have got 46 number of chromosomes. So, it contains the complete set of chromosomes. So, we are diploid, clear, having 46 number of chromosomes, 2N. But when there will be formation of gametes, when there will be formation of gametes, let's say when there is formation of sperm and the egg, the male gamete and the female gamete, the gametes will be haploid. That means they contain half the number of chromosomes. So it will contain 23 number of chromosomes. So gametes will be haploid. Clear? So here also you can say that inside the embryo sac we are saying that there are 7 cells. Though we have counted there are 8 cells, 1, 2, 3, 4, 5, 6, 7, 8, but we have told that, we have learned that the central cells will be considered as a single cell which is formed by the fusion of two nuclei. So therefore the central cell will be diploid, 2N. Alright, because it is formed by the fusion of two nuclei. So generally gametes will be haploid, clear? And after fertilization, when the zygote will be produced, it will be diploid. So keep in mind that the central cells will be diploid, 2N, clear? So first thing we have learned that inside the embryo sac we will find 7 cells, clear? Though there are 8 cells, but the central cells will be considered as a single cell which is formed by the fusion of two nuclei. First point. Second point, let's see now. Distribution of 7 cells. So this particular end is called the micropylar end, and opposite to the micropylar end we will find chalazal end. So in the micropylar end we will find 3 cells, in the chalazal end we will find 3 cells, and in the center we will find 1 cell. The polar nuclei, we call it central cell. We call it central polar nuclei. Clear? So, in the micropylar end, we are finding 3 cells. 1, 2, 3. Clear? Out of the 3 cells in the micropylar end, the 2 cells will be known as synergids. Clear? In the micropylar end, there are 3 cells. Out of the 3 cells, the 2 cells are known as synergids, and 1 cell will be known as the egg cell, the female gamete. Clear? So in the micropylar range we have got 3 cells. Out of the 3 cells, 1 will be the female gamete, that is egg, and the 2 cells, remaining 2 cells are known as synergids, that will help in guiding the pollen grains inside the embryo sac. Now, opposite to micropylar end, we have got chalazal end. There are 3 cells. These 3 cells are known as antipodal cells. Clear? So those 3 cells which are present opposite to the micropylar end, they are known as antipodal cells. Clear? Next one, we have got the large central cell, or we can say the central polar nuclei cells. Clear? So what we have learned? We have learned that inside the embryo sac there will be 7 cells, 3 plus 1 cells. Though in the center there are 2 nuclei, but we will consider it as a single cell. So it will be diploid, 2N. Clear? Now in the micropylar end we will find 3 cells. Out of the 3 cells, 1 will act as the egg, the female gamete, and the 2 are known as the synergids, which will help in guiding the pollen grain towards the embryo sac. Opposite to micropylar end, we will find 3 cells. The 3 cells are known as antipodal cells, and in the center we will find the central cell, or we can find the polar nuclei cell. So this is the structure of an embryo sac. So till now we have learned the structure of pollen grain. Clear? We have learned the structure of the ovule, and we have learned the structure of an embryo sac. Clear? And inside the embryo sac we will find 7 cells. Clear? Okay. Now look at this point. Okay, let's figure. So here you can see stigma, style, and ovary. Here the part of gynoecium. Stigma, style, ovary. Right? This particular structure which you are seeing over here, this is the germinating pollen grains. Clear? This is the germinating pollen grains. So now we are going to talk about the process of fertilization in case of flowers. So here what we learn? We learn about double fertilization. Alright, we learn about double fertilization, and we also learn about the process or the topic called triple fusion. Clear? And also we learn about endosperm. So these three things we will learn under the topic fertilization. So listen very carefully and let's go through the diagram. Clear? So first of all, is the female part of flower having stigma, style and the ovary. And inside the ovary we will find the ovule. So this is the embryo sac. Clear? This is the structure of embryo sac where we will find 7 cells. 1, 2, 3 cells at the micropolar end. 1, 2, 3 antipodal cells. 3 plus 3 will be 6 and the central cell. We call it as 1 cell. So 3 plus 1 will be 7 cells. Clear? So this is the structure of embryo sac. Now we know that the function of stigma is to receive the pollen grain, right? Then pollen tube will be formed. When the pollen grain germinates, pollen tubes will be formed and that pollen tube will run through the style. Clear? And pollen tubes will enter inside the embryo sac. Clear? So now let us discuss in detail about the germination of pollen grain leading to the event of fertilization. Clear? So first of all, when the anther matures, it will rupture and the pollen grains will be released. And that pollen grains will be transferred from the anther to the stigma, which we have learned under pollination. And there are 2 types of pollination: prickle-it, self-pollination and cross-pollination. And for the pollen grain to germinate on the stigma, the pollen grain must belong to the same species, or else it cannot germinate. If the pollen grain belongs to the different species, it cannot germinate. So therefore, the pollen grain that reaches the stigma must belong to the same species, then only it can germinate. Clear? So let's see now. So when the pollen grain reaches the stigma, it will germinate. So pollen grain will germinate. When the pollen grain germinates, there will be formation of pollen tube. Clear? So when the pollen grain germinates, there will be formation of pollen tube, and inside the pollen tube, we have learned that there will be 2 nucleus. We'll draw over here. Okay? So let's say this is the first nucleus, and this one is the second nucleus. So let's consider that this one is the first one is the tube nucleus, and the second nucleus will be our generative nucleus. Clear? So inside the pollen tube, we will find 2 nucleus, tube nucleus and the generative nucleus will again divide to form 2 sperm nuclei. Clear? So generative nucleus will divide to form 2 sperm nuclei. So finally, inside the pollen tube, we will find 2 sperm nuclei and we will find the tube nucleus. Now these 2 sperm nuclei will help in the process of fertilization. So generally, what we find that these particular 2 cells which you can see over here, these are the sperm nuclei, or we can say they are the male gametes. Clear? Now, these pollen tubes enters inside the embryo sac by the help of micropyle. So through the micropyle, these pollen tubes enters inside the embryo sac, and for that, this synergies, the 2 cells at the micropylar end will help in guiding the pollen tube inside the embryo sac. Clear? After that, this tube nucleus will degenerate. Only the 2 sperm nuclei will be present inside the embryo sac. Now, let's see the process. In the first case, alright, in the first case, out of the 2 sperm nuclei, one sperm nuclei will fuse with the egg. Clear? So out of the two sperm nuclei, one will go and fuse with the egg cell, and it will lead to the formation of a zygote, and zygote will be diploid. Clear? Now, second male gamete. Then in the second case, the second sperm nuclei will go and fuse with the central polar nuclei cell. Clear? So the second sperm nuclei will go and fuse with the central polar nuclei, and it will lead to the formation of endosperm. Clear? So one more time. So inside the pollen tube, we are finding two male gametes or the two sperm nuclei. So one of the male gametes will go and fuse with the egg. It will lead to the formation of zygote. And the second male gamete will go and fuse with the central polar nuclei cell and will lead to the formation of endosperm. Now inside the embryo sac, alright, inside the embryo sac, the fusion of gametes occurs twice. There are two times. Why? First male gamete goes in fuse with the egg cell. The second male gamete goes in fuse with the central polar nuclei cell. So the fusion of gametes occurs twice inside the embryo sac. That's why we used to call it double fertilization. Clear? So in double fertilization, the fusion of gamete occurs twice inside the embryo sac. So we'll call it double fertilization. Now what is triple fusion? Triple fusion means, just look over here, right? In the central polar cells, when there was formation of endosperm, second sperm nuclei, we can say one sperm nuclei was fusing with two central polar nuclei cell. Right? We have already specified that inside the embryo sac there are 7 cells. 3 at the micromelar end, 3 at the gelagel end, and 2 nuclei at the center. But these 2 nuclei will be fused and they will form a single central polar nuclei cell. That means in the central polar nuclei cell there are how many nuclei? There are 2 nuclei. So 2 nuclei of the central polar cell and 1 nuclei or 1 nuclei of, let's say, sperm nuclei. Right, so 2 nuclei of the central polar nuclear cell and 1 nuclei of the male gametes, they will fuse to form endosperm. So that means over here there are 3 nuclei which are involved in the formation of endosperm. So therefore we will call it triple fusion. So here the 3 nuclei are involved in the formation of endosperm. Clear? So you have to go to the topic. What is double fertilization and what is triple fusion? Now what is the function of this endosperm, which is formed by the process of triple fusion? This endosperm, it provides nourishment, or we can say nutrients, to the developing embryo. Clear? So after fertilization, there will be formation of zygote, and this zygote will develop into embryo. So embryo will get its nourishment from the endosperm. Clear? Okay. Now next thing, what is the chromosomal number in case of endosperm? We have learned that the gametes are haploid, the zygote will be diploid. But what will be the chromosomal number in case of endosperm? Now endosperm is formed by the fusion of 3 nuclei, right? 1 nuclei coming from the sperm, male gamete, and 2 nuclei from the central polar cell. So therefore, the chromosomal number in case of endosperm will be triploid. It will be 3n. Clear? So zygote will be haploid. Zygote will be diploid, gametes will be haploid, and endosperm will be triploid. Clear? So this is the process or the events of fertilization that occurs in case of flowering plants or the angiosperm. So one more time we have learned that after pollination, when the pollen grain reaches the stigma, it will germinate, it will lead to the formation of the pollen tube, and inside the pollen tube we will find two nucleus, tube nucleus and the generative nucleus. This generative nucleus, it will divide to form the two sperm nuclei. Clear? So it will be in 3 cell states, 1 tube nucleus plus 2 sperm nuclei, so there will be 3 cell states. Now, this pollen tube will enter inside the embryo sac through a point called micropyle. Clear? And these particular two synergies which are present at the micropylar end will help in guiding the pollen tube inside the embryo sac. So when the pollen tube enters inside the embryo sac, this tube nucleus will degenerate. Clear? And the two sperm nuclei or the male gametes will help in fertilization. So out of the two male gametes, one male gamete will go and fuse with the egg cell, leading to the formation of zygote. Second male gamete will go and fuse with the central polar nuclei cell, leading to the formation of endosperm. Now, since inside the embryo sac, the fusion of gametes occurs 2 times, it is called double fertilization. And regarding the formation of endosperm, 3 nuclei are involved. 2 nuclei from the polar central cells and 1 nuclei from the male gamete, which is also called triple fusion. Clear? So remember what is double fertilization and what is triple fusion. Clear? So now after fertilization, there will be changes within the flower, which we'll call it post-fertilization changes. Alright, so let's see what are the changes that occurs in the flower after fertilization. So generally calyx, corolla, they will fall off. They will wither off, they will fall off. The ovary will change into fruit, ovules into seeds, and the wall of the ovary, they will change into the fruit wall, which is called the pericarp. So these are the changes that occurs in the flower after fertilization. So calyx, corolla, they will fall off. Stamens, stigma will also fall off. Alright, ovary will change into fruits, ovules into seeds, and the wall of the ovary, they will change into the fruit wall, which is called the pericarp. And lastly, let's see the changes or the various parts of the ovary after fertilization. So you can see over here, ovary will change into fruit, ovary wall will form the pericarp, the fruit wall, ovule will change into seed, the placenta will change into the stalk of the seeds, the integuments, which will protect the ovules, will change into the seed core, the secondary nucleus will change into endosperm, and the egg cells will change into embryo. And the antipodal cells, they will be disorganized or they will degenerate after fertilization. So here we have finished the chapter pollination and fertilization. Go through the work that I have assigned to you, alright? And for this particular chapter, I will be supplying with the notes, so kindly go through the notes and do your assignment. Till then, be safe, stay safe. Thank you.